This is our acetylcholine signaling and muscle contractions project by Isabella and Sierra. We are video number 27. This is my diagram of the motor neuron, muscle cell, and acetylcholine receptors that are used in muscle contraction. So for a brief introduction, acetylcholine, as represented by these black dots within the synaptic vesicles, is a neurotransmitter used to cause muscle contractions. It opens a ligand-gated ion channels, as seen as these purple trapezoids, that causes a change in concentration of sodium ions, which triggers a pathway that leads to muscle contraction and movement in general. Acetylcholine from the nerves controlling the muscle cell acts as a ligand to find the acetylcholine receptors on the muscle. This binding alters the shape of the receptor protein for sodium ions to move through. Its purpose is to contract smooth muscle, dilate blood vessels, increase bodily secretion, and slow your heart rate. This group of receptor proteins works when acetylcholine reaches the receptors on the membrane. So as you can see, the acetylcholine moves past the synaptic vesicles, down past the voltage-gated Ca2+ channel, and all the way to the receptor. This allows, when acetylcholine reaches the receptors on the membranes of muscle fibers, membrane channels open. Open channels allow an influx of sodium ions into the cytoplasm of the muscle fiber. The sodium influx sends a message within the muscle fiber to trigger a release of stored calcium ions. Stored calcium ions diffuse into the muscle fiber, which causes a contraction. Another diagram I have, the yellow thing is representing a motor cell, the pink is representing the action potential, and the red thing is representing the muscle cell. Again, a motor neuron and muscle cell. Its advantages of acetylcholine in muscle contraction includes that it allows for a rapid response. Say your hand is over a stove and you're not even thinking about it. Your hand isn't just going to stay there. No, it's going to immediately retract. This is an example of an immediate response. This is because of the ligand-gated ion channels, as seen on the surface of the muscle cell. This is also where acetylcholine is present, as it is hydrophilic, polar, and located on the surface of the cell. Its disadvantages, however, acetylcholine is degraded, so it cannot be used again to move sodium ions, and although the response is rapid, it is short-lived.
Now I will explain how acetylcholine causes a muscle contraction response. In this signaling pathway, acetylcholine is the signaling molecule, ligand-gated sodium channels are the receptors, and the other molecules involved are sodium and calcium ions. This is a diagram of a motor neuron containing vesicles that hold acetylcholine. The synaptic knob is depolarized and the charges on the inside and outside are flipped, making the inside positively charged and the outside negatively charged. This opens the voltage-gated calcium ion channels and allows the calcium to diffuse into the motor neuron. The vesicles containing acetylcholine fuse with the membrane of the motor neuron, and acetylcholine is released by exocytosis. The acetylcholine diffuses toward the sodium ion channels and attaches to the binding sites. This causes the sodium channels to open, allowing sodium ions to flow into the muscle cell by facilitated diffusion. The high concentration of sodium ions makes the inside of the muscle fiber positively charged and depolarized, which opens up the calcium channels. Calcium ions diffuse into the muscle fiber where they interact with proteins and cause the muscle to contract. The response is terminated when the sodium channels close and the acetylcholine is released. The acetylcholine then gets broken down into choline and ethanoic acid by an enzyme called acetylcholine esterase. The choline and ethanoic acid are absorbed into the synaptic knob and are recycled into acetylcholine.
Next, we're going to be talking about nicotinic receptors. So say you have a nicotinic receptor, as represented by this purple oval, and you have one key that represents acetylcholine and one key that represents nicotine. Nicotine is an exogenous agonist that is commonly associated with the use of smoking. It is basically a competitive inhibitor of acetylcholine. So, say nicotine binds to the nicotinic acetylcholine receptor. It works like a key in a door, thus why nicotine is represented by a key. Once the nicotine is binded to the receptor, it causes the same response as acetylcholine by opening up sodium ion channels and letting charged ions such as calcium and sodium flow into the cell. You can see that once this is binded, Sodium ions and calcium ions flow into the cell, much like acetylcholine was. However, the difference in concentration leads to feelings many smokers are used to, such as enhanced cognitive ability and reduced anxiety. The signaling pathway is then affected because as nicotine competitively binds to the nicotinic receptors, the binding of the agonist to the nicotinic receptors triggers a conformation change of the architecture of the receptor, which opens the ionic channels during a few milliseconds. The channel is selective for cations, especially sodium. Its opening thus leads to the brief depolarization. Then the channel closes and the receptor transitionally becomes refractory to agonist. This is a state of desensitization, which is represented by the squiggles. As you can see, once this happens, the receptor usually goes back to a state of rest, which means it is closed and does not work anymore. This leads to more receptor levels increasing. As you can see, we now have 4 receptors, and leads to feelings of nicotine addiction and the craving of nicotine. And this is our finished presentation for acetylcholine and muscle contraction.